Yeah, hello. Today we want to talk about the thing that has been trending in Kenya about GMO. So, my fellow people, I want to help you so that you can understand what is GMO, the process of making a plant to be a GMO or goods to be a GMO like maize, beans. What is the process involved in it, so that you make a preoccupied and post-occupied decision about this, what is being told about the GMO. So what is GMO? GMO is just an abbreviation of genetically modified substance. So here we are going to talk about the steps that it takes. It takes 4 steps. One, the first step is to identify. Second, to copy. Third, to insert. And 4, to grow. You copy, paste. So it's like the same thing as you do about the phone. You have identified the message you want to copy, then you copy, and then you paste. As simple as that. So we are going to take an example of maize as one of our subjects. In Kenya, we are going to start with the process. GMO plant step by step. I have said the step 1 is to identify. What is to identify? To identify is, for you to produce a GMO plant, scientists first identify what the trait they want to have. What trait do you want? It's like, I desire a trait in someone. Maybe the guy is cool or the lady is cool. I want that trait. I wasn't born with it, so I want that trait. What should I do? I have to copy that trait to insert in me so that I produce such a trait. So the scientists have to identify that trait from a specific plant. What do I want to produce? I want to produce a maize that is high yielding, in that I get more produce from it. Or I want a plant, or I want a maize that is resistant to drought. I want a plant that is resistant to insects. So what should I do? I have to identify that trait. So the scientists, they will identify that trait, such as resistant to drought, herbicides, or insect. Then they find an organism, e.g. plant, animal, or microorganism that already has that trait within its genes. So I want a plant that is resistant to drought. Then I look for an organism that has that trait of being resistant to drought. Once I identify that, then I get that plant, I get the genes from it. Mind you, the gene is the basic unit. Here we deal with the gene. It's genetically modified. So once I get that genes, I take it, I copy that gene, I insert that gene, then I grow that gene. So once you have copied that genes, in this example, a scientist want to create an insect-resistant corn to reduce the need to spray pesticide. He wants to reduce the cost of production. So I go to the scientist. I want to grow maize in Likoni, or I want to grow maize in Ukambani, I want to grow maize in Mandera. But I want this maize to be resistant to insects so that I reduce the cost of production. They identify a gene in soil bacterium called Bacillus fungus, which produces a natural insecticide. So these scientists have gone to him or her. I want to produce that gene that should be resistant to pests. So they will have to go to the soil. They get that bacteria that is working my head. Then they produce those pesticides which produce the insecticide that has been used for many years, traditional and organic agriculture. So the main thing here is the scientist will go according to your needs. So we have identified So the first step we said it was identify. I have explained about what is identification in GMO process. Now the second thing, now we are going to the second step is you copy. We identified the trait that we want, e.g. Moses has a trait of being fierce. So we want the trait of fierce. We go to the scientist and we tell him, can you produce fairness in Moses? Then he will produce. So now we want to copy that trait. Now after scientist finds the gene which with the desired trait, the trait that we talked, we want a crop that is resistant to drought, they copy that gene. Now what they do, they copy that gene, they go and look for it, then they copy it. For example, the corn, they copied the gene in BT that will provide the insect resistant trait. So we want a maize that is resistant to drought, or we want a maize that is resistant to diseases. Now he'll go and look for a crop that has resistance to diseases to that area. Like in Kitui, we say now in Kitui there is infestation of a certain disease of a crop. Now they look for a gene of that disease itself, then they will copy that disease, so that you can produce a maize that will be resistant to that disease. In case that insects come and eat that plant, he will die. So they will copy that gene, that trait they want for resistance. Now the third step will be insertion. You identify what you want, they copy that gene, now they want to insert the gene. Now next, the scientist uses tools to insert the gene into the DNA of the plant. You see, you identify, you copy, you paste. Now you have to know where you want to insert this text that you have pasted. The same thing with the scientist. Once they copy the gene, now they want to insert the gene. Mind you, the gene is in the DNA. The DNA is the core unit of every cell. Now, by inserting the gene into the DNA of that plant, scientists give it the insect resistance trait. Mind you, the thing that we identified, we want a crop that will be resistant to a certain disease. We look for that gene of that certain disease, so that we produce something that will have the shield of that disease whenever you've been attacked. The same as vaccine. Now, this new trait does not change the other existing trait. You are a drunkard, you are a drunkard, you are a smoker, but in you, we want to insert a trait of being humble. Mind you, it won't change the core behavior of yourself. The only thing, we want to introduce a new trait in you. Then after inserting that in the laboratory, now it is being taken to the laboratory. Mind you, this process involves a lab. Scientists grow the new plant to ensure it has adopted the desired trait, the trait that we have identified in step 1. Now, the desired trait will produce a plant that will be resistant to that disease, either in greenhouses and then in small field. In small field tests before moving it into the large field tests. So it is confined in a small field. In a greenhouse, then we take it to a small field, we test it before we release it to the world. GMO plants grow through in-depth review and safety tests before they are needed to sold to farmers. They have to make sure whatever you are producing is safe to human, whatever you are producing is safe to animals, whatever they are producing is safe to the world, to the environment. So it has to go a vigorous test before being released to the public, before being released to the entire population. At the end of this video, I'll show an algorithm pattern, how this thing goes by. Because there are some some tests have to be done. The PCR test, which is polymerase chain reaction test, to identify, in case this thing turns negative, it is released. If it turns positive, meaning that it has an impact to these people, it's not released. In fact, it's been destroyed in the laboratory. So, before making a judgment, before making a conclusion, you have to understand the process that it has. You have a chicken, but you want this chicken to have a trait of laying more than 30 eggs. We look for that gene from another chick that has such a trait. Then we search into the DNA. Then we produce it in a lab. Then we carry a test. We wait for it to see, will it produce these eggs? Then we test those eggs. Are they harmful to the human being? If you get that point, then comment. Give us a comment and tell, do you still agree with the GMOs, or we are still questioning it? And kindly subscribe for more information. We'll be bringing more news. We'll be educating you. You understand. Subscribe. Thank you.